Most of the research that I've been presenting here is about antibodies that are directed against acute myeloid leukemia. These antibodies we found by searching the B-cell repertoire of patients that cleared their leukemia after allogeneic stem cell transplantation. So what I think is very exciting and interesting about our research is that we did not decide what the target of immunotherapy is, but we investigated what the immune system decided what the target of anti-tumour responses should be. And that's exactly what we did.
Could you tell us more about how this research was conducted?
So what we did was we selected a few patients that had high-risk leukemia, relapsed leukemia, for example, after transplantation, and that nevertheless, after, for example, donor lymphocyte infusion or after a second transplant, cleared their leukemia, and we isolated their B cells and we immortalized these B cells by transfusing them with BCL6 and BCL-XL, and by doing so these B cells are arrested in a stage where they express the B cell receptor on their surface and they produce antibodies, but they don't differentiate any further. So you can do this and you can keep these cells in cultures for a long time, and they produce antibodies that you then can use for investigation. So what we did was we used the supernatant of these B-cell cultures that contains the antibodies and screened that supernatant for binding to target cells of interest, so leukemic cell lines basically, but also blasts from patients. And by doing that, we found quite a number. From 3 patients, we actually found 17 B-cell clones that are specifically directed against leukemia, AML, but did not bind to all sorts of healthy tissues.
Yeah. So you managed to take, I suppose it would be the graft anti-leukemia reaction.
Exactly.
And direct that in a more anti-tumour fashion.
Yeah, exactly. That's what we did. Yeah.
And would these kind of B-cells be maybe a transferable course for therapy if people have this innate reaction to tumors?
Yeah. I think they would, because these are human antibodies. The technology that we used was developed by a spin-off company of the Academic Medical Center in Amsterdam, and the company is called AIM Therapeutics. And by doing the same thing, they also made very interesting anti-RS virus antibodies that are actually now already tested in humans. And from these studies, we know that these human antibodies are safe and can be used for therapy or prevention in other human beings. Yeah.
Is that something that you'd be interested in following up on, or would there be any other steps or paths for this therapy that you'd think would be interesting?
Yeah, I think it would be. So actually we found a bunch of antibodies. One group of antibodies that we found have killing capacity by themselves. So when they bind the leukemic cells, they actually kill leukemic cells without help of other cells, without help of complement. They're really killer antibodies by themselves. So those antibodies are very interesting also to further study the exact mechanism that they use, because of course we could then, if we understand the mechanism, exploit that mechanism perhaps to kill AML cells. But also you could imagine that you use these antibodies as a therapy alone, or perhaps in combination with existing therapies like standard chemotherapy for AML. Other antibodies that we found is also interesting because that antibody is really binding to every leukemic cell that we could get our hands on, and it's really specific for AML. So that would be really a candidate as a naked antibody, like we use rituximab in combination with chemotherapy. In a mouse model, we are testing that now in a mouse model. But also that would be a really interesting candidate to further explore as an antibody-drug conjugate or for CAR T-cell development. And
it sounds like it would complement checkpoint therapy very well as well.
Yeah, definitely, yeah.
Well, that answers all the questions I have prepared. Is there anything else you would like to add?
I think it's exciting, and I think what is interesting, as I said at the beginning, what is interesting about this is that we as researchers did not decide what the target should be. We really exploited what is happening in nature and took that and learned from that, and that really learned us a lot. And through doing that, new targets were exposed. Yeah, and I think that is really what is exciting about this research.